Let's focus here. So number one says you should be able to explain the replication of bacteria cells by binary fission.
Like split in half.
Right. But what's one thing that they have to do before they split in half?
They have to go through the cell cycle.
No. It's something that all cells are going to have to do before they can split in half.
Oh, separate from
They have to copy the chromosome.
Copy the chromosomes. Now, bacteria cells, it's a lot more simple because there's just one chromosome. They got one circular chromosome, they copy it, no big deal, okay? And eukaryotes, because of the nucleus and everything, it gets a lot more complicated. But yeah, binary fission is basically they copy their chromosome and then they just split in half. Easy. Okay? What type of organisms go through binary fission? Yes. Yes to both. Prokaryotes, which are bacteria. Those are the only things that do binary fission.
Wait, what?
Prokaryotes, which are bacteria, are the only things that do binary fission. Okay? All right, listen. Number two, explain the structure of DNA during replication, including chromatids and centromeres. Okay, now I just put chromatids there, but remember we usually call those, what word usually goes in front of it? Sister chromatids. Okay? I'm telling you right now, on the test, there will be a picture of a chromosome. You will have to be able to label what's a sister chromatid, what is the centromere, which one's the P arm, which one's the Q arm. You guys had a homework assignment where you did that, so look over that homework assignment. Okay. What's that?
I didn't. Me, Bryce, Abby were gone. Or maybe Bryce was. Me and Abby were gone, and you told us just to.
Yeah, I was talking to that man.
Oh. Well, you want to look over that. Maybe look at somebody else's. Okay, and remember the centromere is just kind of it's a protein that holds the two sister chromatids together pinched in the middle. Okay?
So,
Isn't diploid too? Diploid as well.
You're getting there.
explain the structure. I mean, that's just the
Well, it says explain the structure of DNA during replication. So that's where it has the chromosome and sister chromatids.
Okay.
Okay? When it's not in replication, then you wouldn't see the sister chromatids. It's all straight. Okay? We're reviewing here. So get out your study guide. We're on to number 3. So number 3 says explain the difference between diploid and haploid cells and how they relate to homologous chromosomes and zygotes. Okay? Here I've got your karyotypes that you guys did. I've got those graded and put in the grade book. So remember, like chromosome number 1 right here, the fact that there's 2 chromosome number 1s, we call that a homologous pair. Each of those chromosomes has, they're kind of identical as far as the types of genes they have on them. Now, they may have slightly different traits, and we'll get to that later, but the whole point is, if this chromosome has genes on it that code for your hair and your eye color, then this one is going to as well. Okay? So they kind of match up as far as that goes. Remember that one of these chromosomes has information that comes from your mother. One of these chromosomes has information that comes from your father. But because there's a pair of them, we call this type of cell diploid. So diploid cells have a pair of each chromosome. Now for humans, there's 23 pairs, so there's 46 chromosomes, but it doesn't matter what organism you're talking about. If it's a diploid cell, there's a pair of each of the chromosomes. Okay? Haploid would just be one of them. So what happens is, and we'll study this in the next chapter, if you have an organism that makes a gamete, so a sperm cell, in each pair, one of these is going to go into one sperm cell, one of them is going to go into another. so that they get separated. So that would be, those would be haploid cells because they only have one of each of the pairs. Okay, so basically when you form your gametes, they get split up. They get split into or into their pairs, or their pairs get split up. So remember, as far as we go, and other mammals, haploid cells are or I should say the only haploid cells are the gametes, sperm and ova. Which part? I haven't talked about the zygote yet. Okay. As far as a zygote goes, remember a zygote is when you have a sperm and an ova that come together. So if you take two haploid cells and you put them together, you get a zygote, which would be haploid and haploid make a diploid. Well, eventually it will grow into a fetus, sure. Okay. So in our whole life cycles, as far as growing up and making babies and all that kind of stuff, the only time we ever have haploid cells are those singular sperm and ova cells. Everything else is diploid. Okay? All right, number 4 says explain the difference between autosomes and sex chromosomes. And I mean, there's not a lot of explaining to do, really, other than to say that we have a bunch of autosomes, 22 pairs of autosomes, and we have one pair of sex chromosomes that determines our secondary sex characteristics. But you should know that females have 2 X chromosomes, generally, and males have 1 X and 1 Y. So for a male, it's a little weird because we call this a homologous pair even though the chromosomes look quite different from each other. But that is still considered a pair, the sex chromosomes that are there. And basically what I want you to know there is that the other chromosomes are called autosomes. So 1 through 22 are autosomes. Pair number 23 is your sex chromosomes. I could ask a question like, how many pairs of autosomes do humans have? Okay. Just one of them. So, remember this, draw it on here so it gets captured. If this is a chromosome, right? Well, that's terrible. Okay, I'm going to try that again. Yeah, it sounds easy, right? Something like that, okay? And it's got the centromere in the middle here. This whole thing would be considered a chromosome. This little bar right here is a sister chromatid. Together, oh yeah, I'm not going to draw that again. Cheat. Together, this is a homologous pair. So a homologous pair is 2 chromosomes. Technically, a homologous pair would be 4 sister chromatids, but only 2 of them are together. Yeah.
Okay.
Yeah. Alma was asking back the question. All right, let's move on, though. Number 5 says you should be able to recognize the karyotype and how it can be used to study a genetic disease. This is a karyotype. You guys should know that. You cut all those things out. But on the test, you're going to have a few different karyotypes, human karyotypes. What you need to do is be able to, number 1, tell if it's a male or a female. Okay?
Is there one where a woman can have 2 X's and a Y?
If you have, so if there's a Y chromosome at all,
It's a guy.
then as far as secondary sex characteristics go, we would consider it to be a male.
So then they can have 2 X's.
You can have 2 X's and a Y, yes. Yeah, but if it's got a Y chromosome at all, then
Okay.
in most cases it's going to turn on those male secondary sex characteristics. Okay? If there's no Y, then it should be female. So what you need to be able to do also, though, is to tell whether the karyotype is normal or if there's an abnormality. Now, I'm not going to ask you, like, name the abnormality as far as whether it's Turner's syndrome or something like that. I'm not going to have you memorize those things. But you should be able to look at a karyotype and go, oh, here's an abnormality, and be able to point it out. Right. Okay. So, I mean, that's not too hard. You just got to look at all the autosomes and make sure there's 2 of each of those. And then you got to look at the sex chromosomes and make sure there's either 2 X's or 1 X or 1 Y. Otherwise, we would consider that to be abnormal.
Okay, what do you say, like, if there's more of the autosomes, you say trisomy, how do you say if there's more X's?
I mean, we don't have a generic term like that, but, I mean, like I say, you're just going to be asked to point it out.
Oh, like
If it's,
symbol?
right.
We have to like speak
I mean, you can. You don't have to. Okay. 6. Should we explain the different phases of the cell cycle and how they relate to a cell's life cycle? Okay. So remember, at first, we kind of have 2 general groupings. There's interphase, and then there's the mitotic phase. Not mitosis, that's different. The mitotic phase does contain mitosis, but those things are kind of separate. Not separate, but they're different levels of classification, we should say. Okay? Now, if we're comparing interphase and the mitotic phase, which one does the cell spend most of its lifetime in? Interphase. And you should be able to break down interphase and talk about the 3 parts of it and what happens in each of those parts. So what would be the first part of interphase? G1. Growth 1 is the way I would put it. And what happens in growth 1? Yeah, normal growth and development. Doing what a cell's supposed to do. Okay, what happens after G1? And what happens in S? Copy the chromosomes. They copy the DNA. That's where the sister chromatids are made. Okay? We're used to seeing chromosomes look like this because that's how we see them under the microscope, because the only time we can see them under the microscope is during mitosis. But in a cell's normal life, like G1, homologous chromosomes just look like this. It's during the S phase that the cell then copies those, and you get the sister chromatids to make up the other part of what we call the chromosome. So that copying happens during S. Remember, S stands for synthesis. Synthesis. Synthesis means to make. Just means to make something. Synthesis just means to make. Period. End of sentence. Okay? Yeah, then G2 is the next part, and then G2 is to make organelles in preparation to go through mitosis and cytokinesis. That stands for growth 2, except in this case, the growth that's happening is the cell does get a little bit bigger, so it has more cytoplasm and stuff, but really the growth is the growing of extra organelles. Okay? It's kind of like before you move out to college, you're going to have to buy extra stuff that you used to using of your parents, like food. I was thinking along the terms of like dishwasher or laundry soap and that kind of stuff. Maybe a dryer, that kind of thing. But regardless, you're going to have to buy some extra stuff to be able to move out on your own because you're used to using it. So the cells are doing the same thing. The cells are using all this stuff, but now there's going to be 2 cells. There's got to be enough for both of them to get along.
Actually, if I was going to college, I'd rather have a sheet.
Okay. We're getting there. We should finish, but let's push on here.
Wait, we didn't talk about.
Well, yeah, we'll come back to that because the mitotic phase includes mitosis and cytokinesis, and so we'll get that down here. Probably would have made more sense to put those up there in the numbering order, but here we are. Number 7 says, explain what cancer is.
It's like when the cells stop growing.
Or they like split really fast.
It's mutated cells.
Right, all that stuff. So cells are mutated in a way that they continuously grow and develop. And remember, there's a couple other things that make cancer cells weird. Number 1, they don't care how crowded they get, they just keep growing. Most cells, if they kind of fill up a space, they're like, nah, that's enough. But cancer cells don't give a dang. And remember, the other thing that makes cancer cells weird, what? They're immortal, for the most part. They don't really have a kind of a scheduled lifetime like most cells do. Most cells, they go through a certain number of divisions and they're dead. But cancer cells, they just keep going. Okay? And that's really, for this test, that's the most that we need to know about cancer. Okay, number 8 says, explain the different stages of mitosis, be able to recognize them based on their structure. Now, when we were doing the microscope stuff over the last couple of days, it seemed like when I'd ask people, what stage is that under there, there was struggling to even come up with the name of a single phase. Remember PMAT, right?
Yeah.
Okay? Prophase,
Anaphase.
PMAT, metaphase, anaphase, and telophase. Okay? On the test, there will be pictures of some of those phases that you may have to put in the right order. Like I said before, you may have some where one of them is missing. You have to determine which one.
Don't tell me.
You would maybe have to draw the one that is missing to the best of your ability. Telophase. Telo means at the end. T-E-L-O. And then phase.
Telo is silent.
Some people do pronounce it telophase, but not me. So, the other thing is we should know some of the things that happen during each of those stages. So we need to run through these quickly, but remember, in prophase, the nucleus breaks down and the spindle is created. Spindle fibers attach to the centrioles at one end and the centromeres on the chromosomes. Okay? All right, Bryce, what happens during metaphase? Chromosomes all line up, and the spindle fibers are attached to, each chromosome has 2 spindle fibers attached to it, running to the centrioles on either side of the cell. Right? Spindle fibers are connected to each side of the centromere, and there's one running off each side to the centrioles at either end of the cell. Okay? Anaphase. The sister chromatids break apart and move to opposite ends of the cell.
They break it down.
They do break down the spindle fibers as they go along. That's true. Okay. And then in telophase, spindle finishes breaking down, so like any spindle fibers that were still connected to the sister chromatids and the centrioles, even though they're short, will all break apart so that the centrioles and the chromosomes are not connected anymore. That's a long way of just saying the spindle finishes breaking down. The nucleus starts to reform around the chromosomes, and the cytoplasm starts to pinch, getting ready for cytokinesis. Okay? Definitely look at those structures to make sure that you got them in the right order in your head, as far as how they look. And then number 9 says explain the difference in cytokinesis between plant and animal cells.
Cleavage, furrow, and plants have the cell plate.
Cell plate, right. Now the cell plate, remember, is what will end up becoming the cell wall between the two cells. But until it's completely built, we call it the cell plate. And then, yeah, animal cells just have the cleavage furrow, which is basically just, hey, the cytoplasm is pinching together in this area, and there's kind of a rift until they finally come apart. Okay? Now, one last thing that I want to make sure that you guys focus on. What is the purpose of mitosis?
To split. Well, they don't split the cell.
Not to
Make
split
new
the cell.
cells.
Not to make new cells.
Oh,
To split the chromosomes. To separate the chromosomes. Okay? Yeah, you can say DNA in there. Yeah, to separate the DNA, that'd be fine. But remember, the point of mitosis is not to make 2 new cells. It's just a part of the overall process of making 2 new cells. Okay? The whole point of that, just that section of mitosis, is only to separate the chromosomes. Cytokinesis, that's where the cells actually split. It is separate, not the same thing as mitosis. It comes afterwards. Okay? All right, those of you that missed the first little bit there, I have been recording this. I will put it on the website, link to YouTube channels. I'll send out a remind with the link to it too, but it might be a good thing to go back and look at Sunday night.
Make them in Monday.